Okay, so welcome back after the coffee break. So we will continue with Nick Barton, who's at the ICTS in Austria. Nick has done spectacular work on many disciplines. He has worked empirically and has never lost interest in connecting some things, but he's mainly a theoretician. So you wouldn't consider yourself? Anyway, let's see after the talk.
Okay, thanks, Christian. Yeah, I wasn't sure when people asked earlier who was an experimentalist, but I do field work on hybrid zones, so there are no real experiments there. It's more observation. Anyway, yeah, it's a pleasure to be back here. I was last here, I think, 6 or 7 years ago, and it's just the same, except the trees have grown bigger, and so that's nice. So I hope that I can build on the introduction of yesterday and from Bruce this morning to really, in this lecture and the following lectures, to try and emphasize and explain the relation between population genetics, which is probably the background that most of you have more strongly, population genetics with quantitative genetics. And what I'll start is by, actually, so I should start with some logistical things. I think I'm down to give a tutorial or some exercises on Thursday afternoon. And I put a couple of exercises on the Slack channel, I think, wherever that is. So do have a look at those beforehand, but the idea is that we'll go through them together in the class. Okay, so we'll go through a couple of exercises, which should be possible to do in your heads or with pencil and paper, so no big computational requirements. Okay. So where to start? I think with Darwin. Always start with Darwin. So his theory of evolution by natural selection really emphasized the importance of gradual variation, continuously varying traits. And that was for two reasons. One is that he thought evolution was a very slow, gradual process. Larger. Yes, thanks. How big? Another boost? Okay, yes. I mean, the notes here are actually on the Slack channel, and I don't think you necessarily need to read them. I hope that I'll say everything that's on here. They're there just to orient you in case you want to take a little nap, and then you can wake up and see where you are, and so on. Okay, so Darwin emphasizing slight variations. And his theory was, or at least his argument for evolution, for what he called descent with modification, was accepted remarkably quickly. And partly, I think, because it gave a very powerful framework for conducting all kinds of different biological research, for bringing together work on embryology, paleontology, physiology, and so on. So it was enormously stimulating to biology. And really dominated late 19th century biology and through to the present. But really quite surprisingly, and probably quite disappointingly to Darwin, the central importance of natural selection as the mechanism for adaptation, as we now accept, was not understood, and was not understood even by Darwin's champions like Huxley, Galton, and so on. So there are various reasons for this. One was, and really quite an important one, was pointed out by Fleming-Jenkin early on in a review of the origin of species, and that was that under the current understanding of inheritance, which was thought to be a kind of blending process, where not unreasonably two parents would produce offspring who are a mixture of whatever came from those parents. And that mixing process would necessarily reduce variance. And in fact, the calculation showing that the variance produced by this mechanism of blending would decrease by a factor of 2 every generation. So the variance would decrease by a factor of 2 every generation and would be dissipated very quickly. And that was actually one of the first, if you like, population or quantitative genetic calculations done in the early 1870s by Davis, I think was the first person to point this out. So this was a real problem because Darwin spent much of the time in the origin giving the evidence that there was abundant heritable variation for the kind of traits that matter, and this was the material for natural selection, and yet it should be dissipated very quickly, and he had to just say, well, we don't understand this, and this was a big gap in the theory. So that was, of course, a stimulus for work on understanding inheritance. And of course, there was also the practical application at this time. Agricultural innovation was really important in making sure that the growing cities in the Industrial Revolution were fed. And so agricultural innovation, practical horticulture and so on was driving a great interest in understanding inheritance. And yet it seemed to be quite obscure. The most important, from our point of view, the most important sort of research program was called later biometry, founded really by Francis Galton, who developed the first statistical methods for describing continuous variation, ideas of regression and so on, and compiling the first big data sets on heritable variation, comparing parents and offspring, not just for height, but for, to us, rather strange characteristics, intelligence, earning power, and so on. And infamously founding and propounding eugenics. So this biometric program was continuing, was developed not in a very prominent way, but it was developed through the late 19th century by Carl Pearson, a very powerful, prominent mathematician, developing a lot of multivariate statistical methods. Ralph Weldon, his colleague, a zoologist who did some of the first experiments measuring natural selection. And they were motivated really by trying to understand how natural selection would work and thinking that understanding how distributions changed under selection would give us some information about that. So this biometric program was sort of steadily rumbling along, not a very large program. And of course, with hindsight, we say, well, why weren't Mendel's ideas picked up? Mendel working at about the same time as Darwin, publishing in sort of 1860 around then, and yet his work being neglected until 1900, when it was discovered by 3 independent research groups who were doing experiments trying to understand inheritance, hit on more or less Mendel's laws and discovered it had been done 40 years before. So at that time, in principle, many of the problems with Darwin's ideas could have been resolved, but in fact, there was a bitter controversy between the biometricians emphasizing Darwinian ideas, selection on slight variations, inheritance of continuously varying traits, and so on, and much more prominently, the new young geneticists. I was going to say classical geneticists. Now we see it's classical genetics. Then it was the new cutting-edge research. And those geneticists thought, well, what matters are major mutations. That's what we're studying. Therefore, that's what must be important. And they really denigrated the importance of continuous variation and said it was just noise, irrelevant. And there's a very nice account of this in Will Provian's book, Origins of Theoretical Population Genetics, and many other interesting historical accounts of this time. It's really quite, it's quite disconcerting how personal animosities in the end and feuds between different research communities can slow down science by decades. And so the resolution really only came with primarily Fisher's 1918 paper, which explained exactly how the combined effects of multiple Mendelian variants could together produce normally distributed variation. And I mean, amazingly, in this paper, he introduced analysis of variance, the really powerful statistical technique, with the purpose of dividing this variance, trait variance, up into genetic and non-genetic components and so on. And so this we see as the foundational work, establishing quantitative genetics. There were other ways in which the resolution was achieved, partly through just careful selection experiments showing that selection on continuous traits really did work, unlike, despite the arguments of the geneticists. So by the 1920s, population genetics, more or less as we know it, was being developed by Fisher, Haldane, Wright, and so on. Encouraging for young scientists, yes, very encouraging, because he submitted it to the Proceedings of the Royal Society, who rejected it. And I think there was, do you remember these quotes, Bruce, from yes.
Crowe said it was too, so Pearson was one of the reviewers, yeah. It was too Mendelian for the biometricians
Yeah. And I think one of the quotes from the biologist said that we biologists have not much use for this kind of thing at the moment. It was just, all this calculations. And it's still a pretty opaque paper. So, yeah. So in principle, there was a resolution. In fact, that resolution could have come 20 years earlier. In fact, even Mendel had put in a little cryptic comment that, well, continuous variation could be due to the combined effects of multiple Mendelian factors. And people like Bateson, who coined the term genetics, who was one of the leading early geneticists, had sort of hinted at this. And yet somehow all this was lost in a great battle between different schools of thought. So things were resolved, but nevertheless, there was a continuing separation between quantitative genetics and population genetics. Population genetics developing into what we call the evolutionary synthesis, the modern synthesis, the synthesis of Mendel with Darwin, but concentrating on traits which were inherited in a simple Mendelian way, chromosome variation, pigment variation, and so on, and developed, taken into the field by people like E.B. Ford and Dobzhansky, particularly in the US. That population genetic tradition really developed quite separately from the quantitative genetic tradition, which was developed with a practical application in mind by the breeding industry, by Saul Wright, for example, who was working for the US Department of Agriculture, developing methods for quantifying inbreeding and so on. And so there was actually, by the 1950s and 60s, quite a sophisticated statistical framework for explaining how continuously varying traits could be inherited, how they'd respond to selection, and really very impressive computational methods, particularly given that there were no computers at this time, or at least there were only human computers. So there was a coming together of quantitative genetics and evolutionary genetics in the 1960s and 70s through people like Alan Robertson, Russ Lande, Michael Bulmer, who saw that the quantitative genetic ideas were actually relevant to a lot of evolutionary questions. And there was quite a flourishing of papers, particularly from Russ Lande, I think, asking really evolutionary questions in terms of quantitative genetics. And that led to what's now called evolutionary quantitative genetics, but it's become a kind of a subfield, and there's still quite a separation, as there is a fragmentation of the field into many different sort of sub-communities. So what I want to do, just before I actually get into the substance of the talk, is to say that this separation of fields, which is more a social phenomenon than anything, also corresponds to very different views of evolution. So if you think in a quantitative genetics framework, then you take the basic observation, which is really quite a remarkable one, and I think it was alluded to in one of the talks we had yesterday, that pretty much any trait in any organism, at least any sexual outcrossing organism, will show high heritability. In other words, a large fraction of variance of order 20% to 60, 70%, around half of the variation will be due to genetic differences. Okay? And that ubiquity, suggests that selection will be able to act to change any trait to drive it to its optimal value pretty quickly. And of course, that's our empirical observation. If we select on a population, we will get a continuing response, more or less as long as we have the patience to keep selecting. This became particularly apparent, I think, through work of Ken Webber. A lot of early selection experiments were limited by the feebleness of research students, I think. So somehow their advisors could only get them to select on a few hundred flies for a few tens of generations before they were exhausted. But these experiments would then reach some sort of limit, some kind of plateau, and I'll come back to this later. But Ken Webber was this kind of very interesting character who liked making machines for large-scale selection, and I'll mention one of these later. So he was able to do selection experiments on a large scale, thousands, tens of thousands of individuals, and found what was kind of understood, but with rather little direct evidence, that if you select on a large population, you'll be able to get further. You'll have more initial variation. You'll be able to continue for longer without losing that variation through inbreeding. And so selection on large populations is really effective. And so this whole idea really reinforces the worldview that actually evolution should be shaped by selection largely, not limited by lack of genetic variability. And so one sees evolution as an optimizing process. And then, there are large branches of evolutionary biology which sadly manage to do quite well without genetics, without even reading Walsh and Lynch or Lynch and Walsh. But one can get a long way simply by thinking, how do selective forces shape organisms, thinking of it as an optimization process. So that's the view I think one comes to if you come from a quantitative genetic background. And I mean, another way of looking at this is to say that the enormous success of plant and animal breeding, I think has been driven largely independent of our detailed scientific understanding of the genetic process. It's been driven largely through the 20th century anyway, by simply systematically measuring accurately and collecting information and being rigorous about how you run a selection program. So that's one view. But if we then turn to the population genetics view, I'll sort of describe what I think now dominates at least the molecular end of the field, and that's what Lewontin called the classical view. He contrasted in his book, 1974, The Genetic Basis of Evolutionary Change, very well written, very accessible summary of the state of things at the time, just after the abundance of molecular variation had been discovered by Lewontin and Hubby, amongst others. And just at the beginning of this fierce battle between neutralists and selectionists about what maintained this variation. So at any rate, Lewontin emphasized what he called the classical view, in which it's very simple. There's at any one time in any particular environment, there's a best genotype, and variation is largely due to deleterious mutations driving organisms away from that optimal state. And so variation, initially under the classical view, would be due to deleterious mutation, very occasionally due to beneficial mutations coming in, sweeping through the population, as they sweep from low frequency to high frequency, contributing a little bit of variation, but not very much. Adaptation consisting of successive substitutions. Kimura in 1968 put forward the neutral theory, which said, well, actually a lot of molecular variation is just neutral, irrelevant, but one can see this as an extension to the classical view. It's really in the same framework. And I think this way of looking at things, the idea that evolution consists of successive sweeps with variation within populations being really somewhat uninteresting, that kind of dominates the way we look at sequence variation now. It's the simplest way, it's the place to start. But it really, I think, has meant that the alternative view, which Lord called the balance view, associated with Jain in particular, that has become much less prominent, although still, much more influential, I guess, in the more ecological genetic side of things, if you actually work with real organisms. And so on that view, variation in a population is much more interesting. There isn't a single best genotype. There are many, many genotypes which are maintained by various kinds of balancing selection, overdominance being the simplest one, but various kinds of fluctuating selection, spatial heterogeneity, and so on. And this, I guess, was stimulated, for example, by Jain's study of chromosome inversions in Drosophila, where large chunks of chromosome are inverted and therefore held apart, kept distinct from their homologs, because the different arrangements can't recombine with each other, and therefore able to diverge and adapt to different niches. And there were really striking early observations in the 30s showing that different inversion types would increase and decrease through the seasons, suggesting they were adapted to different seasonal habitats. So we have these very different views, but now I think we almost always work, at least in the parts of evolutionary biology that really are focusing on understanding sequence variation. We start by thinking, can we find selective sweeps? Can we understand the nature of background selection due to deleterious mutations? These processes are very much in the classical mold. And maybe I'll come back to that next week, but I think there's also a parallel with the dominant view of molecular biology, where one thinks of manipulating pathways, detecting function, using major disruptions, using knockouts and seeing what happens, mapping out the connections between different molecules, the way they regulate each other, producing, a relatively straightforward and discrete picture in which one doesn't really attempt to make a quantitative model of variation in gene expression and interaction in a quantitative way, partly because that's very hard. Again, it's the place one would start. One starts by looking at the simplest possible explanation. But I think there's a bias in that way of looking at things, which some ways parallels the sort of biases that Bruce was talking about earlier, that we can find, if we have relatively crude techniques, we find major effects. Is it that those are really how things are operating? Anyway. Okay, so that was a lot of rather general background. Just to finish this waffling by pointing out some difficulties, again, which I think we'll see in various different places during the whole workshop. Under the quantitative genetic view, I mean, it's clear that there is heritable variation for many traits, for almost all the traits that we try and measure. But then if we think how many traits are actually needed to describe an organism, more important, how many traits are needed to be specified to allow that organism to survive and reproduce. So clearly, if you try and describe the morphology, the physiology, the behavior of an organism, there's an infinity of variables. Most of those may be kind of irrelevant. Who knows? But if you try and add up even the most basic things, you get to an enormous number. If you think of, say, gene expression, you think of the expression of 20, 30,000 genes, but that expression has to be at the right time, at the right place, in the right environment. So you rapidly get to very big numbers, even just looking at that level. And so then you sort of worry quite a lot about whether all of those things can be under strong optimizing selection, because if there is heritable variation for all of these myriads of traits, hundreds of thousands of different traits, then you imagine that organisms are losing fitness for this enormous number of reasons. And how do they keep functioning? And this is part of the puzzle, I guess, also of how it is that organisms can reliably develop from a single egg to a complex multicellular organism with elaborate behaviors and so on and so forth, with a relatively small genome. And I guess most people, students, are too young to remember the shock we had when the human genome was sequenced, and we only had 22,000 genes. It was embarrassing. Drosophila had 14,000. I mean, Drosophila is a magnificent organism. It deserves 14,000. But still, there were bets beforehand that there would be 100,000, 150,000, 200,000 genes. The number that came in was much, much lower than people expected, I think because we had a grand view of our own complexity. But it really is a puzzle as to, on the one hand, how so few genes can code for a complex organism, but on the other hand, how so many genes are maintained by the rather erratic and stochastic process of natural selection, by what Herschel called in the 19th century in a derisive term, the law of the higgledy-piggledy. Somehow it works. So that's the problem with the quantitative genetic view. How can so many traits evolve reliably? If we turn to the classical view, I mean, to me, it just seems a bit dull. Maybe this is just a prejudice. But there's not much going on. There's a limited number of substitutions going through, which contribute a bit of variation, a bit of heritable variation in fitness. But that's limited by the number of substitutions we see in the genome. And the rate of molecular evolution, the rate of coding evolution is really quite slow. And you can also bound the number of deleterious mutations. The total mutation rate in the human genome is 50 to 100. It's a pretty large number. Most of that is hopefully neutral. The rate of deleterious mutation is worryingly high. Maybe it's 1, 2, 3. It's larger than 1. It's actually unclear how we can maintain ourselves despite that high rate of deleterious mutation. But still, it doesn't contribute that much fitness variance. The fitness variance is of order total genomic mutation rate times selection coefficient. And since we think selection coefficients on deleterious mutations are not that high, that's not a big number. So that's kind of fine, unless, like me, you're trying to measure fitness variance in the wild, and then you get depressed if you think it's only 1%, and then you'll never be able to measure it. But it's also disturbing because most of our theories, all of our theories for how, for example, sex and recombination can be maintained, require a high level of fitness variance. They all require some means by which basically modifiers that can increase the rate of sex, increase the rate of recombination, can be gaining an advantage through facilitating natural selection. And those are the dominant theories. And the theory kind of, I think, is reasonably well understood. But the big problem is that they seem to require too much fitness variance to be compatible with a rather boring classical view. So maybe there's more going on. And finally, under the balance view, which I find just attractive because it makes for more interesting theories and more interesting calculations and so on. But actually, that's a problem because genetic variance then is maintained by all kinds of idiosyncratic processes of fluctuating selection, of spatial variation and so on. And yet we see ubiquitous variation. We see similar levels of heritability in cages of flies in a really boring environment versus in natural populations. So that's also hard to understand. So anyway, I'm just trying to bear in mind these kinds of difficulties which we have. We have, we can get carried away with the sophistication of our technology and our statistics and the size of our textbooks and so on, but there are still fundamental problems in the field. So, having tried to warm up there, what I'll do is basically today and in the next lecture, which I think is on next week sometime, I'll talk primarily about directional selection. And I'll be talking about the infinitesimal model, which has been mentioned a couple of times before, and I'll try to define that firstly at the phenotypic level without, almost without any reference to discrete genes, and then asking how that relates to the genetic level. So the themes actually throughout all the lectures I'll give is, what is the relation between quantitative genetics and population genetics? How do we see these two different ways of looking at things, how they're connected? I'll also, towards the end today, start talking about limits to the response to directional selection, firstly in terms of the infinitesimal model, and then more broadly. Okay, any questions? Not that I've said anything very substantial yet. Okay. So, yes.
Mutual variation we know is lesser than what we expect from the response. Right? So the mutual genetic variation is much smaller than what we expect. And on the other hand, there is a lot of variation.
So why do you say neutral variation is less than we'd expect?
Paradox,
sorry?
Lewontin's paradox.
Ah, okay, yes.
Yes.
Yeah. Yes.
Which is smaller than what we expect.
Yes. Yes.
And here,
no, I was talking about interesting variation here, actually. I mean, in the sense of not neutral, right? So, of course, neutral variations are our primary source of information about how things work, what the demography is, and so on and so forth. But I was really thinking of variation that affects fitness and about the material for selection. And trying to contrast the view, I think one has, if you come from quantitative genetics, where you think there's variation in everything and everything can just evolve to its optimal and we can select on any trait and improve crop yields indefinitely, et cetera, et cetera. On the other hand, under the classical view, there's relatively less variation when you're looking at individual sites or individual genes. You're looking at the genetic level then. And of course, mutation rates are very low. And substitutions seem to be relatively rare because we can see directly the rate of amino acid substitution on a phylogeny, and that rate is of order the mutation rate. Partly some of that is neutral, but selected substitutions also are not that common. So we have a contrast in the amount of variation, and but I'm focusing on the variation that matters in the sense of matters for adaptation, because we're supposed to be talking about polygenic adaptation. That's it. Okay. Well, no, I was trying to come up with three different kind of worldviews one could have. You could have a quantitative genetics view, you could have this classical view, which I think is well understood by people. And I was just trying to mention the balance view because I started out as a student in 1976 and Lawrence's book had just come out and I've really enjoyed reading it. And he was putting this, very big contrast between these two worldviews. And it's really the classical view that's taken off now. And I think actually, if you work, as you do, in more sort of ecological genetics and so on at the level of the whole organism, there are many, many interesting phenomena which really are not captured by that. And where one, thinks about how different behavioral strategies can be maintained in a population, different adaptation to different niches, etc. All of those things are much more comfortable in the balance view. You're interested in the in the way in which heterogeneity and variation in fitness can be maintained. I suppose.
The focus is what's maintained.
Yes, I think the average quantitative geneticist just wants variation to be there, wants to understand the structure of that variation so as to get a selection response. That's my caricature of the thing. I mean, it's all a bit of a caricature. So, yeah.
Where's the red queen fit in terms of groups?
Balance. Number 3, I would say. Yes. I mean, I guess it's non-equilibrium. It's a way in which variation can be maintained and rates of evolution can be much faster than they would be if it's just waiting for a beneficial mutation, which is the kind of simple view you'd have under a classical sort of model. Okay, maybe I should say what the red queen is for people. This is quite a way back in the 1960s, I think, when Wayland came up with this. And it's just the idea that, why doesn't evolution just evolve to the optimal state and that's it? The ideal organism sits there and that's it. It's because other organisms are evolving, and the red queen is taken from Lewis Carroll, and it's that I can't remember the story exactly, but she has to keep running faster and faster to keep in the same place, and you have to keep running faster and faster in order to to keep up with your competitors. And actually, we can see that almost directly in this. I rather like Alex Kondratov's middle-class neighborhood experiment, where he just took cages of flies and turned off selection. How do you turn off selection? You just take 2 flies and you produce 2 offspring and 2 offspring and 2 offspring. He was a bit disgruntled living in suburbia in the US. He thought that's just 2 parents, 2 kids, keeps going. No selection, right? And so then you see that the viability and fecundity reduced by 1 or 2% per generation, which he interpreted as due to deleterious mutation, but one could do similar things in a more natural setting, and then you would also have the effects of competition and coevolution and so on. Okay, so the infinitesimal model. And this, I only started to get interested in this when, for a reason I can't quite remember, I got together with Alison Etheridge and Amandine Weber to write a sort of review and some sort of mathematical justification for the infinitesimal model. And I thought this was classical part of quantitative genetics, which in a sense it is. It is the actual basis of almost all the quantitative genetics that's done in practice. But the term has very obscure origins. It's associated with Fisher. People say Fisher's infinitesimal model. And it maybe refers to a passage in his 1918 paper. In fact, Alison and I argued about which passage it was, but anyway, there's a section in which he shows that in the limit of a very large number of genes of small effect, the additive genetic variants will change very slowly under selection, and I'll go through that derivation very quick, very sort of a very outlined way. But Fisher didn't use the word infinitesimal, and the word started to be used by Michael Bulmer and others around the sort of 1970 time, and I asked Bill Hill where the term came from, and he said, I don't remember. Asked Michael Bulmer, and I asked Michael Bulmer, and he said, I don't remember. We just all called it the infinitesimal model. So this sort of word emerged at some stage, and people kind of knew what it was. It's kind of a bit disconcerting that something can be lost to living memory in a relatively short time. I actually trace it back to a paper by Galton, Typical Laws of Heredity in Nature in 1877, where he, in a very sort of aristocratic Victorian way, did quite a big breeding experiment by taking packets of sweet pea seeds and posting them to his friends and giving them instructions. And then they presumably got their gardeners to do the crosses, and then he collected the seeds. And what he was interested in was in the variance in seed size in the offspring of plants that had parents that had different seed sizes. And he said, it's a nice quote, I was certainly astonished to find the family variability of the produce of the little seeds to be equal to that of the big ones. And so it was, and I thankfully accept the fact, for if it had been otherwise, I cannot imagine from theoretical considerations how the problem could be solved. What he was actually referring to in the problem was something tucked away in the appendix of that paper, where he does a calculation of, as far as I can make out, of the equilibrium genetic variance under stabilizing selection, under a model in which when you cross 2 parents, you get offspring with some variability. And that is the infinitesimal model, as I'll explain in a bit. And so he did actually this calculation, which then wasn't done for another 100 years, or 90 years, actually, when Michael Bulmer did the calculation, without, I think, noticing that Galton had done it. Anyway, what Galton's alluding to here is that he was really trying to find laws of inheritance. And he had in mind that if the variance of the distribution of the offspring was kind of arbitrary and depended idiosyncratically on, what the parent values were in a very complicated way, it would all kind of be hopelessly complicated. But he found there's this very simple pattern, which was that the offspring were normally distributed. He was very struck by as many people were, by the ubiquity of this Gaussian distribution, and that variance was independent of the values of the parents. They could be small, or they could be big, or whatever. So he then thought, well, we can actually start to do calculations. So the model, the infinitesimal model, defined at the phenotypic level is very simple. It just says that every individual has, now I'm using modern terminology now, but we have to think of the breeding value of an individual. And this is a separation that wasn't understood by Galton. That was a major obstacle to actually understanding inheritance. The idea that you can break up the phenotype into non-genetic and genetic causes. And in fact, I think it's important to realize that this is actually a concrete, observable thing. You can take an individual, any one of you, and you can measure your genotypic value very simply by just cloning yourself and ensuring that all these clones are then reared under the same conditions as you were reared in the same environment. So, well, that doesn't work for humans, but for many organisms, of course, you can do that. You can raise genetically identical individuals, and the genotypic value is simply the average value of the trait for that genotype. And of course, it depends on the environment. Actually, I'll be talking here about the breeding value, which is slightly different. Most of what I'll say is assuming additivity, so then in that case, they're the same, but the breeding value is something also quite concrete. You take an individual, you cross it at random to other members of the population, and you measure the average of the offspring. And then you take the deviation of those offspring from the mean, and you double it on the grounds that you are one of the two parents. So the breeding value is defined as twice the deviation of offspring produced from random mating from the mean, right? So again, we'll come back to quantify this a bit better, but each individual then has this breeding value. Right? And what you say is that if you take two individuals with breeding values Z1 and Z2, the offspring have breeding values at the midpoint, but with a normal distribution around that, with a fixed variance, V0, which is simply a constant of nature. It's a constant of that population. It's kind of fixed in the simplest version. And crucially, that is independent of the parents' values. That's what Galton was observing. And so with this model, you find that very quickly, you take these two parents, and you could take any two parents, just two individuals, they'll produce offspring with some distribution, with the variance V0. If you then randomly mate these individuals, you'll get pairs of parents with different average values. And so the variance actually now will be V0 over 2, which is the variance of the midpoints of two randomly chosen individuals from this. The variance of the mean of two values is simply half the variance. So this is the variance of the midpoint, plus you get an extra variance V0 coming from this infinitesimal model. And so you keep doing that, and actually pretty quickly you get to an equilibrium where the variance is twice this within family variance. And that equilibrium, what's happening is that when you cross two individuals, the variance of the midpoint is half the variance in the population. So that if we say that the variance at time t plus 1 is equal to the variance at time t divided by 2, because we're crossing the two, that's the blending inheritance. This is the calculation Davis did in 1871. But crucially, we add this extra variance V0, and so we'll rapidly get to a Gaussian distribution with this. Okay, so super simple. So what is random? I mean, I'm assuming random mating, yes. Yeah. Absolutely. I mean, you can obviously, and in fact, I think Galton did this. You can also say, suppose there's assortative mating, that will increase the variance. And actually, under this model, kind of is not really intuitive. If you have enough assortative mating, the variance can keep increasing indefinitely. But I'll stick to random mating. Okay, so that's the absolute basic model, and it can be stated empirically without any reference to genes. And of course, now we know where this variance comes from. It's not mutation. That's very, very, very much smaller, come to that. But it's released by recombination. And the underlying assumption, at least which reconciles this infinitesimal model with Mendelian inheritance, is that we have large numbers of genes of small effect, which are unlinked. Okay? And so that means that when you have, let's say one of the diploid parents has two chromosomes which cross over and they produce gametes, which are a random combination, a random selection of the genes on one or other genome, that generates variability. And we'll come to sort of work through the calculation of that variability. So the variance is released by the segregation of different genes that are heterozygous in the parental individuals. If we think of diploids, it's released at meiosis, essentially. And so we actually have to modify the infinitesimal model by allowing for the fact that the parents might be related. And this is important in practice because we're often dealing with a pedigree with some artificially selected population, which is of limited size. And so the variance released is actually equal to V naught, this constant, times 1 minus F, where F, we've seen before, is the probability of identity by descent of the genes that are coming together just before meiosis. So if we think of a haploid system, maybe it's easiest to start with that scheme. We can define it. Maybe I shouldn't even, I shouldn't be drawing chromosomes because we're supposed to be having unlinked genes, so lots and lots of genes on lots of different chromosomes. These are haploid parents, and they come together and produce, maybe I should, I think we have the technology here, don't we? Colors. These are genes coming from different parents. So this is a transient diploid which immediately produces offspring with different combinations of genes, and they might have, mostly from this parent, they might have mostly from this parent, or they'll most likely have a mixture of the two. Okay. So the variability is due to segregation in meiosis. And this F now, in this haploid case, refers to the probability of identity between the two homologous genes. I should have drawn these as homologous, I guess. If these are actually inherited from the same parent earlier in the pedigree, then no variation is released. I shouldn't say heterozygous. We're really talking about identity by descent. Are they inherited from the same gene earlier in the pedigree? To the extent that they are, they don't count. So we only want the fraction of gene pairs which are non-identical in meiosis. If we have a diploid system, now we have is a diploid, and another diploid parent. Now we have F1, which is the probability of identity between two genes in this parent, and that can be calculated from the ancestry of this. And we have F2, which is the probability of identity of two genes here. They produce gametes, right, do a little. Two gametes, and then they unite and produce a new diploid, or set of diploid offspring. And the variance is now 1 minus F1 plus F2. It's the average probability of identity of the genes coming from this parent and from this parent. Okay. But either way, actually we can define the infinitesimal model without even knowing about ploidy. just in terms of the probability of identity, really, between the genes that are going into meiosis. Okay, so that's really the bulk of the infinitesimal model. We've got the process of inheritance, of random segregation, which is the process of recombination here. If we have a simple situation where we have a well-mixed population of effective size NE, then the probability of identity, the heterozygosity, let's say 1 minus F, the probability of non-identity decreases by a factor of 1 minus 1 over 2n per generation. So that's the sort of simplest kind of Wright-Fisher model. If we have the ideal Wright-Fisher model, the factor by which non-identity decreases, and therefore this segregation variance within families decreases, is 1 minus 1 over 2n. And then other models would have a different rate of decrease, and we describe that by this 1 over 2ne in effective population size. And if we have a detailed pedigree, we can actually calculate this in quite a lot of detail. So the final thing we could throw in, and this is a little bit harder to justify mathematically, you have to make some assumptions about the nature of mutation, but empirically, and this is something Mike Lynch has spent a lot of time on, you can measure the rate at which genetic variance is generated by mutation simply by taking genetically identical individuals and either just letting them accumulate variability and measuring the additive variance for a trait through time. It takes a long time, but we've done, or selecting on the population and waiting for a selection response due to new mutations. And what's really kind of quite striking and surprising and puzzling is that one finds relatively high rates of increase of mutational variance at the trait level. And of order 10 to the minus 3, or even more than that, up to 10 to the minus 2, implying that the heritability of a population is regenerated after a complete bottleneck over hundreds or 1,000 generations, over a short time scale, relatively short time scale. So this, I think we've now understood. It was probably more striking when this first emerged, but we now understand this as simply being due to the very, very large numbers of genes, each of which has a small mutation rate, but if we have thousands of genes involved, then we have a high rate of input of mutational variance. Okay, so that's the basic infinitesimal model. How am I doing for time? So what I want to do next is go through how we relate that, how we justify it based on a very simple genetic model. And I'll start with an additive model. In fact, I'll probably finish with an additive model. I don't really want to go too much further than that. But before getting into this additive model, I want to actually make a statement, which is something that we only, I think, really worked through in this paper with Alison Etheridge and Ameline Weber, that one can justify the infinitesimal model even beyond additivity. That traditionally it was understood as, well, the limit of many, many genes of small additive effect. And that's the simplest way to think about it. And maybe that's most of what's going on. But the way I like to think about it is that it's really a consequence of the fact that the range of variability of traits within a population is much, much narrower than what could be realized. And we kind of know that empirically, because if we take a population and select up or select down, we can go a long way. We can go many, many tens of standard deviations in the order of hundreds of generations. Right? And that's why we're well fed. It's why agriculture works. So if you take the additive view, then down here we have the most negative possible individual. Up here we have the most positive possible individual. And if we were dealing with the whole range of variation, then crossing this individual with another of the same would produce no variability in the offspring, because they'd have the same genotype. There'd be no generation of variation by recombination. So knowing the phenotype tells you about the genotype and therefore tells you about how much variance is going to be released. Whereas if you take individuals in the middle, you can generate a lot of variability because there are many, many combinations. And the point is that if you have this kind of additive model and you kind of throw the genes together at random, the variance will be quite limited relative to the total range. So that's another way of stating the observation, that if we select on this population, we can actually get a long way outside the original range. So that's an intuition based on an additive scheme. But actually, I don't think one has to think of it quite in a strictly additive way. What we're really saying is that there's lots of genotypes here, which all give a similar kind of phenotype, somewhere near something that works, something that functions, maybe some kind of optimum. We don't have to have strict additivity. We just have to have a situation in which knowing that an individual had this phenotype or this phenotype doesn't tell us very much about the genotype. So it simply requires a large degree of redundancy. And in fact, in the paper, well, a couple of papers we've had now on this, we show that you can extend the infinitesimal model to certain kinds of epistasis and to dominance, right? It gets much more complicated, so I'm not going to go into it any further, because you have to then say, well, we have to track all the variance components. We do have additive variance, we have non-additive variance, we have dominance variance, but one has predictions from classical quantitative genetics for how those variance components change as a result of inbreeding. You have to then track all the multi-way inbreeding coefficients, which means you just have to look up which chapter? Anyway, you look up the correct chapter in Walsh and Lynch and you get the answer. Maybe Lynch and Walsh, I can't remember. Anyway, I remember digging into this. There are many, many coefficients. Yeah, okay. So,
there is, as you point out, there's a delicate balance between getting a bounded dominance variation and bounded inbreeding.
yes.
And that's really delicate.
Yes, yeah. And that's, to me, a sort of empirical question. Why is it that we get reasonable inbreeding depression? But I'll mention that a bit later. I'll come back to that a bit, yeah. Anyway, I was just trying to sort of warm up to the very simple-minded additive model because I wanted to defend the infinitesimal by saying that it doesn't require strict additivity. I think a lot of let's say molecular biologists and non-evolutionary biologists are very critical of the whole of quantitative genetics because they say it assumes additivity. I am not the sum of my genes. It's obviously a stupid model. And it is obviously a stupid, unrealistic model, and yet it gets us a long way. And it's really, I think, also still quite puzzling that that's true. Okay, so just some notation then. I'll keep things really as simple as possible by saying that we have a number of loci, say little n loci, and they have two alternative alleles, which we label 0 or 1. So we just have this indicator, big X, which is 0 or 1. And so we say that the trait value of an individual and we'll assume a diploid individual, and we'll just say that it's the sum of the effects of the one copy from the one parent plus the other copy from the other parent, so there's no dominance, no interaction, just the sum of the effects at that locus, and then summed over all the loci, and they can have different effects. The effects of adding one allele, replacing a 0 by a 1 is alpha I. And so we can have an allele frequency pi equals the expectation, average in the population of xi, it's kind of obvious. And we can say that the mean of the trait is then just a sum over the allele frequencies times the allelic effects times 2 because we've got diploids. And then we can say what's the variance, and it'll be additive because the whole model is additive. What's the additive genetic variance? Well, it's equal to the sum over the loci. And I'm going to make two crucial assumptions here, and I'll come back to these a bit later. Let's assume to begin with that the states of the genes are independent both between the two parents, that is, the two copies inherited from different parents are independent of each other, and they're independent between different genes. So the XI and the XJ are independent, and we call that linkage equilibrium, and Bruce referred to that earlier. Come back to that. So assuming independence then, we just have a sum of alpha I squared times twice the variance, well, I'll spell it out, times the variance of xi plus the variance of xi star. And so we can make another assumption that the distribution of effects inherited from the one parent is the same as from the other parent. I mean, that needn't be true, but let's assume the simplest thing. So this is just equal to twice the sum of the square of the effect. So we're dealing with variance, so mean squared deviation, times the variance of xi. And the variance of these indicators, and this is just something really useful to know in population genetics, this is really a distribution with 0 or 1, 0 with probability q, and 1 with probability p. So the mean is at p. And so what you have is a fraction q who have a deviation 0 minus p. So the squared deviation is 0 minus p squared. We add p, 1 minus p squared. That's fraction p that are value 1, and therefore a deviation from the mean of 1 minus p. So you put all that together and you get p times q with a bit of algebra. Okay? So this is just something worth remembering. The variance of a two-state variable is just p times q. So we have okay, so we've got that. So that's your basic formula for the additive genetic variance. And just a comment now, which is really crucial to the whole thing, which is that if we say that this VA is of order 1, in other words, it's fixed, it's what we observe, then as the number of loci increases, the alphas have to scale with 1 over the square root of the number of loci, because they're adding up over n things. Each of them is contributing alpha squared to the variance. So the alphas have to decrease in magnitude by square root of number of loci. And that's really where the infinitesimal model comes from. So I'll just briefly ask now, what's the variance amongst offspring? Because that's really where we started out. We asked if you take two parents with different values and you cross them, what's the variance amongst the offspring? And that's quite easy to work out because you've got a whole lot of genes. Let's say, let's think of a single locus. We can add up the contributions from all the loci. We're assuming they're independent. So if you have two parents who are homozygous, then the offspring will have all the same genotype, right? So if parents are homozygous for, let's say, big A, and the other one's little a, then the offspring are all big A, little a, there's no variation, right? So we've got a fraction, let's say 1 minus 2pq with Hardy-Weinberg. That's the probability that a parent is homozygous. So I could write that as p squared plus q squared, but I'll write it like this. The probability they're both homozygous is that, and then they contribute nothing to the variance amongst offspring. With a probability 2 times 2pq times 1 minus 2pq, we're going to have one heterozygous parent, and so it'll contribute either a naught or an alpha, okay, with probability 50%. And so the variance contributed is alpha squared over 4, because it's a binomial variable with equal frequencies half to a half, either 0 or alpha. And so the variance of effect is alpha squared over 4. I mean, another way to see it is if you've got equal probabilities, then you're deviating either by minus alpha over 2 or plus alpha over 2. It's the contribution from that parent. So the mean squared deviation is alpha squared over 4. And then finally, we have the possibility that both parents are heterozygous, in which you get alpha squared over 2, because they're both contributing variance. And so we just add all those up, and I hope we get alpha squared pq 1 minus 2pq plus alpha squared times, yes, 2pq squared, and that cancels with that, and amazing, we get a contribution alpha squared pq. So if we sum that up over all the loci, then this within family variance v naught is equal to sum over loci alpha I squared pi qi, which by a miracle is half the additive variance, okay? So there were probably quicker ways to get to that, but that's just confirming that if you have, this additive model, in fact, we don't have to assume alleles of smaller effect or anything in particular, the within family variance is half variance, additive variance in the whole population. Okay, so next, next we can ask, well, how does selection affect a population under this very simple model? So, yep. Yep. So, which assumption? Ah, okay. So I think I made lots of assumptions actually all the way through. Yes. Okay, so I think I was making assumptions at various points. One of the assumptions I made was in calculating this additive genetic variance, and I will come back to that. That was the assumption of independence between genes at different loci linkage equilibrium and between the genes inherited from the two different parents. So that will affect the additive variance.
Just only the quick answer.
Yeah.
Is that going to just change the factor of 2 or something more is going to happen?
It'll be more complicated. Yeah. So I'll come to that. It will. Don't worry. It will get more complicated. Yeah. Okay. So, where was I? So I think we've seen some of the ingredients here before. So we want to look at the effect of selection, and it's easiest to think of selection, at least for me thinking in terms of natural selection, by thinking of the relationship between fitness and trait, right? And if it's a multiplicative or an exponential relationship, we get a curve a bit like that. One can also think in terms of the relation between the mean fitness and the mean of the trait for a given variance, and we'll get some similar curve. In fact, the mean fitness is equal to the sort of average of the distribution, which is some sort of Gaussian with a certain mean and variance, times WZ, Z, and so on, and so forth. So one can think of these two. For purposes of this talk, for directional selection, one doesn't have to worry about the distinction, but there is a distinction. I'll be thinking in terms of the selection gradient, which is the gradient of log mean fitness with respect to the mean, which often is approximately this, give or take some corrections. So this is just saying how much does the mean fitness of the population change when we increase the mean of the population. And the selection coefficient on an allele, this is a result from Sewell Wright, is given by the gradient of log mean fitness with respect to change in allele frequency. Now, what's the relationship between selection gradient at the level of the trait and the selection acting on a particular allele with a particular effect? Well, we know that this can be rewritten as just partial derivatives, ignoring influences of change of allele frequency on the trait variance and so on, but I'll just simplify it here. So then we know that the rate of change of mean fitness with respect to pi, sorry, this should be a half for diploids. This is just equal to 2 alpha I. Because if we change the allele frequency, if we substitute one allele for the other in a diploid, we're substituting 2 copies, and they each have effect alpha I. So this is just then giving us a half, well, the 2's cancel, and we just have beta alpha I, which is kind of obvious in a way. So if we have a selection gradient acting on the phenotype, that's here, and if we have an allele that has effect alpha I, for swapping one allele for the other, then the selection coefficient on that allele is just the product of those two. That's really easy. So that gives us a way of working out the change. I'll come over here to be fair to people. No left-right bias, I suppose. So we have the change in the mean is equal to twice the sum over all the loci of the change in the allele frequency. And we know that if we have a selection coefficient S, the change in allele frequency is SPQ, standard pop gen. So we have now 2 sigma alpha I SPQ, but we said that S was equal to beta times alpha times PI QI. And then we get beta I times so we get another of these quantitative genetic miracles that the change in the mean only depends on the complicated genetic architecture through the additive variance, right? So that's a super simplification. And of course, one can derive this directly by thinking about the infinitesimal model and not worrying about genetics at all. So that's the change in the mean. And the thing that I'll do more or less the last thing today is to now show you how you justify, in a sort of outline, the infinitesimal model in this simple case by asking what's the change in the genetic variance? Because the sort of key observation, the observation that Galton made, is that regardless of the value of the parents, the variance amongst the offspring is constant. And therefore, if you select, you'll get differences in mean, you'll shift the mean, more or less as you want, but the variance within families will stay constant, okay, because it's independent of the value of the parents. So what we have to show is that under this additive model, the change in the additive variance is small in some kind of limit with some assumptions. Okay, so this is actually really easy because we've got the A was by that. And then we ask, suppose we make a change in the allele frequencies due to selection, the change that, which is that. So I'll assume that that change is small. And so then we have this as twice the sum of alpha I squared. And if we just take the gradient of that, well, we don't even have to do that. We'll just write it out, P plus dP, Q minus dP is approximately PQ plus delta P, Q minus P. Actually, and we could keep that, but it's a pain, so we'll cross that out, right? Because ADP is small. So we have here now QI minus PI delta PI, and then we substitute for the change in allele frequency, and we end up with so what I've done there is write in this is alpha times beta times PQ. So we get an alpha cubed, we get a beta, which we can take outside, and we get a PQ in there. Okay? And we can actually write that as, that hasn't really achieved anything, except that I've written S as twice the sum which is actually the skew of the distribution. If you work out the third moment, right, which is the, actually, we have this two-value distribution. This is just looking at the contribution of one gene, right, which has value 0, probability Q, 1 with probability P, then the third moment, the skew, is equal to probability Q, it's 0 minus P cubed, plus probability P, 1 minus P cubed, and that equals PQ Q minus P, I hope. Yeah. So, and then because we're assuming independence, we can sum that up over the whole thing. So the third moment of this distribution of breeding values, this skew, is this thing. And this is just a bit of an aside, but there is a whole machinery one can develop in which you can predict not just the change in the mean of a trait, but the change in the variance and the third moments in terms of the mean and the variance and the third moment. So there's a rather beautiful mathematical way of describing changes in trait distributions in terms of the various moments. Yeah? Yeah, I was just about to say, I was about to say it's all completely useless because it's not, yes, it's a beautiful machinery. The third moment depends on the fourth moment, and the fourth moment on the fifth moment. And I did waste many months or even years of my life trying to close this series. But, you know, you can close it in different ways, you get different answers, but it's really not a good way to go, yeah. But I'm just saying there is this relationship between the moments, right? In this context, the crucial point is to see that the change in additive variance due to selection is going to be small simply because the skew is small if we have the sum of many, many independent effects. That's central limit theorem, right? So the distribution of the sum of many, many things, each of which has this sort of funny bimodal distribution, that's going to be close to Gaussian, and the skew will tend to 0 as 1 over root n. Another way to look at it is to say that this can be written like that. If we think of a sort of funny kind of averaging, so the contribution of a gene to the genetic variance is alpha squared PQ. So we could rewrite this as twice the sum of the alpha squared PQ, which is its contribution to genetic variance, times alpha Q minus P. So this crucial skew, which is telling us how much the selection is going to change the genetic variance, this is really a kind of weighted average of the allelic effect times the difference of allele frequencies. And that will typically be small, actually for two reasons. One, this kind of central limit reason, which is that these alphas, if there are a lot of them, they must be small, of order 1 over root n for given genetic variances. But also because typically, unless there's something biased going on, which there may have been some directional selection or whatever, but other things being equal, these Q's and P's will tend to cancel out. They'll sometimes be positive and sometimes negative. So this cancellation also makes this typically rather small. But we'll come back to the skew, I think, maybe in the last lecture. Anyway, this is just sort of an outline of an argument that under the additive model, selection will change the mean substantially, but it will not change the variance appreciably if there are a very large number of genes. Right? So that's the key justification of the infinitesimal model. Okay. Yep. Yes.
So in that case, we expect this offspring distribution's variance will be different.
It will change, yes. And it clearly must, because if you imagine that you've got a limited number of genes, a limited number of possible genotypes, as you shift towards, you know, the one best genotype, then you lose variation and the variances must shrink. Yeah. Okay, yeah. What, now or later? No, later. Okay, good. Good. Yeah. Okay. Okay, I want to come back to these. Sorry, yeah.
So, when we do see a measurable change in variance of the function number of loci, that's something accessible.
It would be very cruel to propose this. Yeah. I think it's not a sensible project. I mean, it's one of these things one could do as a thought experiment, But if you imagine, I think Bruce mentioned, estimating variances is really hard. You need a much bigger sample size than estimating means. There are not many good, accurate measurements of genetic variance. I mean, there are obviously lots of work on this. And of course, we don't know the number of loci. One could, you could imagine, I'm going down a bad route here, but you could imagine an experiment where you took your base population and then you could have put it through bottlenecks, so you lost variation at some fraction. You could estimate the number of loci which was segregating. You could actually then cross different populations and make sure they all started at the same allele frequency, 50-50. So, there are various schemes you could concoct populations with known allele frequencies and known numbers of loci. Maybe Christian could do this, yes. And then you could start selecting and saying, how does the variance change? And you have predictions. But I just don't think it's a sensible thing to do. I think it would be heroic, yeah. It's one of these things that's observable in theory, but not in practice. Okay, so I made this assumption before about loci being independent. And actually, that was not a very good assumption, because under the infinitesimal model, I should change this. Politics is coming in. Selection can make large changes to the distribution. So I don't know if it occurred to anyone, but everything I've said now is deeply implausible, because I was kind of implying that selection doesn't change the additive genetic variance. But you can just go to a population and pull out the big ones and pull out the small ones, and you've increased the variance, right? And you've increased the genetic variance, right? So you will have increased the additive genetic variance by doing that. So what I'm saying is all contradictory. And to understand that, we have to understand the contribution of linkage to disequilibrium. So let's think then about the top. We start with a population which has a nice Gaussian distribution with some variance. It's sitting happily there, and you apply really strong disruptive selection. So the dashed curve is some fitness. You've applied this fitness function. It's a bimodal strong disruptive selection, and you'll have produced then, if we multiply this dashed curve, the fitness function, by the base population, that gives you the distribution afterwards. It's the probability distribution of the population times the fitness function divided by the mean gives you the new distribution. That's it. And that's the red curve. Sorry, that's the blue curve, I should say. So the red curve has been pulled apart by this disruptive selection. And now it's got this weird shape. It's got an increased variance. What's going on? Well, you apply one round of infinitesimal reproduction. You have random mating. The blue ones sort of mate. So left with left with right with right, so they mate at random. And each of those matings produces a Gaussian distribution of offspring. So what you end up with is a mixture of Gaussians, and mixtures of Gaussians really pull quickly towards something that looks pretty Gaussian. So this is a true calculation. Can't remember how I did it, but this blue curve is not quite, but it's very close to Gaussian. So the thing is pulled back to a Gaussian with a slightly inflated variance. So over one generation, you have, by selection, produce something completely ludicrous with a very high variance. One round of infinitesimal reproduction gives you a new variance, which is still inflated, a distribution which is closer to Gaussian, not quite Gaussian. So what's going on? Why is it seeming to contradict this calculation? Well, what I was doing in calculating this additive genetic variance there was calculating what's called the genic component. Come off. And that is the component there, VA, is twice the sum of alpha squared times PQ. That's contributed by heterozygosity at individual loci. But if you work through the actual genetic variance, you find that it involves also contributions from linkage to disequilibrium. So what we have are individuals with value Z equals some okay. So that the variance of Z, the variance of the breeding values, is going to be the sum over all pairs of individuals, Ci times Zj. So what I'm doing here Well, yeah, no, I should do this more carefully. We want the variance, getting carried on. We want to calculate the variance of this, right? But we're not going to assume that these things are independent of each other. So we write down the variance of Z as equal to the expectation of Z minus Z bar squared. That's the definition of the variance, okay? And so that we can obtain by subtracting the mean from this, right? So the deviation of each individual from the mean is given by x plus x star minus twice the allele frequency. Or we could write that actually in terms of these zeta things, where zeta is equal to x minus p. So these are just deviations of our indicator, either 0 minus p or 1 minus p. And so now we have the sum over all pairs of loci, alpha I, alpha j, times the expectation of zeta I plus zeta I star, zeta j plus zeta j star. And I will still assume random mating. So I'll assume that the genes inherited from the different parents are independent, and I'll assume that the two parents are equivalent. So this is going to be equal twice the sum over all pairs of alpha I, alpha j expectation. In other words, if you look at this product, there's 4 terms. 2 of those terms are correlations between genes in different parents, this and this. And so I'm saying those are 0, the parents are independent. But there's 2 terms, this with this and this with this, which should be equal. But we can then separate this into a sum over single loci, where these are at the same point, expectation. And that's just the variance of contribution of one locus. And then we've got this other term, which has actually many more terms. It's all the correlations between different loci. And in fact, if you work through it, you find that this covariance between the states of 2 different genes is the linkage disequilibrium between them. It's another way of defining linkage disequilibrium, the same as equivalent to the one Bruce gave. Whereas this variance of effects of one gene is just PIQI. So what we have is that this is VA, and this is, I don't know, what's the notation? VLD or VA brackets? Okay, I will change it. I'll have a fluctuating, I'll call it VALD. This does have a stable notation. It's the genic component. So the additive genetic variance has 2 parts. One is the genic component, which stays kind of fairly constant through time, and to which my derivation applies and explains that constancy. And it has another part due to LD, which is much more transient, which you can expand and contract and manipulate, but it dissipates very quickly because these genes are assumed to be unlinked. So I shall finish and give you hope for lunch. It's all hope. By just mentioning, just to scare you really, dominance and epistasis, right? So as I mentioned before, actually you can justify the infinitesimal model more generally than just from the additive case. You can't have arbitrary epistasis because one could easily concoct a model where we had Z evolving under the infinitesimal model, and then I could think, well, suppose I redefine a trait Z squared, or Z to the 10, or some arbitrary functions of Z. Then it's just not going to behave in the same way, because if we cross 2 individuals on the Z scale, they produce offspring which on average are halfway in between. But if we now look at Z squared, then these are going to be something like that. They're no longer going to be at the midpoint. It won't be a Gaussian. It'll be just different. So we can produce counterexamples. But these counterexamples are kind of a bit extreme in the sense that they require a certain systematic epistasis. If you write this out in terms of the pairwise epistasis, all the epistasis has to point in the same direction. And so there's basically a condition on epistasis being not systematic in some sense. And given that, the infinitesimal model still applies. And it's still a bit more complicated because you actually want to then model not just the additive component, but the additive biadditive components and so on. Same sort of thing applies with dominance. It's even more of a nightmare. As Bruce mentioned, you have to worry about what the dominance effects are at each locus, because that determines not just the dominance component of the variance, but the inbreeding depression, both of which are measurable. And those actually play a separate role. In fact, it turns out there are about 5 different coefficients you have to follow, and the whole thing is a bit of a nightmare. But you can do it if you're sufficiently masochistic. Anyway, this is just to finish with a picture, which is a simulation of a model with dominance, not, there we are. And partly I'm just putting this up to show you the kind of variability you get in the LD component. Is there a pointer somewhere? Or a stick? A laser, yeah. Okay. Okay. That's good, it's high powered. Yep. So this is just an example with 1,000 loci. Each of the loci has complete dominance, so there's a completely recessive allele, etc. But the dominance points in different directions for different loci, and the values are rigged so that the additive variance is 0.27, the dominance variance is 0.063. This is actually worth noting because even in a model where at every locus there's complete dominance in the sense that 2 of the genotypes are identical and the other one is different, it's still, with reasonable allele frequencies, you still get most of the variance being explained by the additive component. That's typical. And the inbreeding depression in this particular example is 0.53, whatever. That's the rate of decrease of the trait with the level of inbreeding F. And little population, 30 individuals evolved for 50 generations, and I tracked the full pedigree and then ran 300 replicates on the same pedigree. So the black lines here correspond to the total genotypic value, and the blue line, the additive component, which is most of it. And this shows one of the replicates diddling about. This is the mean values, the overall total trait value, and the dominance component, which is quite small. And the right-hand thing shows, again, for one replicate, the variance components, total genetic variance, the additive variance, the dominance variance, and this other one, which is the purple line here. Red is the dominance variance, purple is the covariance between the additive and the dominance effect. So there's some covariance between them. Those don't change very much. The one point to note is that there's a lot of fluctuation in these total variances. And that's almost all due to LD. The dashed lines show the genic components of these things, the components you get by ignoring the LD. And in fact, the population will relax back to that. It's always pulled back to that. And it's just drift generating random LD, producing noise around that. If we average over all the replicates, we get drift to the mean, but the variance components actually are close on average to the genic component, and that genic component matches actually the theoretical expectation more or less exactly, okay? Because it's a contrived kind of model. So really, the point of putting this up is to show that one can do calculations with dominance. I'm not recommending it, I think, because you have to follow all these multi-way identity coefficients. But more important, if you try and measure actual variances, actual genetic variances in a real population, you will get most of the variability being due to transient LD, which is yet another reason why it's hard to measure these things accurately. Okay, so I have 2 minutes to spare. I'll stop now. Any questions?
I mean, getting that LD to balance is tricky because scales is square root of n as opposed to 1 over n. So you have to
You mean the inbreeding depression you're talking
Yeah.
about? Yes,
Yeah,
yes.
ID, sorry.
ID, yeah, it is. Yes.
The fraction has to become increasingly smaller as
Yes.
n gets bigger in the right way.
So is it then a mystery as to why the divine forces, or whatever they are, have rigged things so that inbreeding depression is, of order 1, and dominance variance is small? I mean, it's a bit of a mystery to me what we would expect those to be. Because I agree, in doing the simulation, I had to jiggle the parameters to get a reasonable value for those two, which is telling us something. Don't know what. Okay, so next time it'll be limits to selection, starting with the infinitesimal and then going a little bit beyond.
And one of the most underappreciated results of Polygenic provinces.
That's what I'm doing. Yes.
So, you were saying stasis, you still have the variance of time size that's independent of the mean, but presumably varies more.
It's kind of actually gets a bit complicated. With epistasis, you have a component, a random component peculiar to the family, and then you have random variation between the offspring within the family. So in the additive case, given the two parental values, the distribution is always centered at the same point. But with epistasis, different families can have different center points. So that's a complication. I'm not sure that I know how to answer whether it's more or less variable under drift. You have all, yeah. I mean, I'm not.
The variance, the variance, the offspring.
Yes, that happens. Yes, and that's an extra thing to keep track of. Yeah, you have two distributions to keep track of. Don't know. Okay, it's probably lunchtime.
What